Now, these questions are now for Professor Pfeiffer. And the first question goes as follows. Can you explain what distinguishes this frequency range from others in the context of biomedical applications?
Yes, thank you for the question. Now, if you want to answer this question, you need to compare it to the currently applied techniques in the optical or visual range for tumor margin identification. And the issue in the visual range is that one needs to stain the tissue to differentiate between cancerous tissue and healthy tissue. And this is done by a histopathology step where the staining, of course, takes time. And therefore, it would be very nice to come up with some faster technique that could be done independently of staining. And therefore, we think we could characterize the material properties at a different frequency range, like the terahertz range. And at terahertz frequencies, you could measure the permittivity, which is the dielectric constant of the material, and use that as a differentiator between healthy and cancerous tissue. And the nice thing about terahertz, of course, is that the wavelength is rather short compared to microwaves, and therefore you get still some reasonable good resolution laterally. And in the near field, of course, as I presented in my presentation, you also are able to get very high resolution in the near field. And therefore you have basically that additional kind of technique that supports the histopathology doctor and is an addition to the visual inspection that you may apply later on. So in this sense, I hope that explains and answered the question.
Now, is there clinical evidence or validation supporting the effectiveness of this tumor identification procedure?
Well, of course, that is a step that we are now looking at as part of the Terahertz NRE project, where we are actually working together with medical doctors and histopathologies. And therefore, we are providing the data that's necessary to have enough statistical results to answer that question. Of course, it's normally a bit of a chicken and egg problem. You know, what's first? Either you have an effective procedure and you are trying to build a sensor for it, or it's the opposite, where you're trying to develop a sensor technology first, and then you want to validate the effectiveness of the technique. So in this sense, we have the sensors now ready, and as part of the Terahertz NRE project, we are kind of now in this phase to really prove that this technique can be an add-on to the existing visual inspection techniques.
And now, can you tell us what is the current technology readiness level, or the TRL, of these sensors?
Yes, of course. Now we are working at the university, of course, and it's not, of course, our task to develop a final product, that's for sure. But we are, of course, using standard microelectronics techniques. So we use silicon chips, which are the same as the ones that are typically run in your cell phone. And therefore, the technology is very mature, basically. And therefore, we consider the technology readiness level to be rather high already, in the level of 3 to 4. But again, as the second question just pointed out, we have to, of course, show the effectiveness of the technique. And therefore, this project is very relevant now to provide that proof, and that brings us a lot closer to real life applications in the future.
And lastly, to wrap this up, what do you consider are the upcoming milestones or the next steps for the Terahertz NRW project in this area?
Yes, thank you for that question. And of course, as I pointed out, we need to get in touch with collaborators on the medical side, and therefore we need to provide at least sufficient samples so that we can feed them with the hardware that they are in need. So we, as a next step, basically we have to build a lot more sensors, and we also have to build evaluation boards so that we can provide these medical doctors with the hardware and that they can run the tests themselves That's going to be a very crucial step as part of the Terahertz NAE project. And this is not something we can run on our own. And we, of course, don't have enough personnel to really travel around and try enough tissues to different patients and so on. And therefore, I think that Terahertz NAE project is very crucial, and it gives us sufficient support to develop enough sensors to run these tests and clinical trials in the future.
So we just received a question on our email, and that goes also to Professor Pfeiffer. Does the detection refer specifically to superficial skin cancer, or were other types of tissue also affected by cancer examined?
Well, at the moment, we are looking into tumor margin identification because that allows us to measure the contrast between healthy tissue and cancerous tissue in one sensor image. So it's, of course, very difficult to acquire just some numbers on a certain tissue and also on different kind of cancer tissues, of course. And the reason is that you have a lot of variations from patient to patient. There's a lot of variation in terms of temperature, for instance. So, you know, a single measurement on a certain cancer type or so is not going to be sufficient to identify that cancer tissue uniquely. Therefore, you always have to measure this in contrast to other cells. And therefore, I think our technique is very useful because we are really able to implement thousands and hundreds of sensors operating all in parallel. And those sensors will provide us with a real image. And therefore, we can see the contrast between certain you know, margins between these different kind of tissues. No? So that is kind of the key here, that we are not only just relying on a certain specific cancer type or something like that. We always see that in combination with other measurements that we do in parallel.